All right, welcome to part one of chapter 6, where we're going to look at reproduction at the cellular level. In this part, we'll look at asexual cellular reproduction in general, and then specifically how it occurs in eukaryotic cells like yours. There's two types of cellular reproduction, and chapter 6 focuses on mitosis or binary fission in prokaryotic cells. But this is asexual, and it's designed to produce daughter cells that are identical to the parent cell. Okay, so this is going to maintain the number of chromosomes. That's going to stay the same. And this is how we grow. This is how we develop. And this is how we replace, we repair. We do replace any damaged or dead cells. Meiosis is sexual reproduction. We'll cover that in chapter 7. In meiosis, we're cutting the chromosome number in half, and we'll see why that has to occur in chapter 7. If we look at mitosis, this is in a sea urchin, and the extreme far left in A, you're seeing a single cell, the fertilized cell or the single parent cell that is now dividing in half to form two identical cells. And then those two divide to form 4, and then those 4 divide to form 8, and then those 8 divide to form 16. And so B, the image B, is showing you that same cluster of cells after 4 rounds of cell division. And again, under ideal circumstances, these cells are identical to each other unless some form of a mutation has occurred. And for a long time, they're identical to each other until we get to a level of development where cells begin to differentiate. And what that means is they start to take on whatever their mature adult cell role is going to be. And if we look at the sea urchin on the far right, you can see that there are some very different structures on the outside that we can see. And certainly there's going to be different structures on the inside. When the cells that form the spines on the outside, or the ectoderm, which is the kind of yellowish or orange beigeish tissue you can see underneath, or any of the organs or organ systems inside, they all at one point started from identical cells. But at some point in time, genes kick in and they start to develop into the mature cell type they're going to be. Same thing happened in you. You started from a single cell that implanted in the uterine wall and divided, and then divided again, divided again, and divided again. And for a long time, all of your cells were identical. It isn't until much later in development that cells begin to differentiate. Some become the cells that will become tissue of the heart or of the brain or of the nerve system or bones or muscles, all the other organs and organ systems and tissues that make up you. But when it started, they came from a bunch of identical cells. Now, we want to control mitosis. We don't want a cell to divide unless we need it to divide in our bodies, and the same is true for all organisms. Sometimes this is going to depend on the environment. If you're a single-celled organism, you're not going to waste energy on cell division if there aren't enough nutrients to support 2 cells where you currently have 1. And in our bodies, we don't want to copy cells if we don't have a need for that cell. So we're going to control this by genes, and we're going to see, in chapter 8 extensively, and then even more so in chapter 9, all cellular activity is controlled by genes, which are pieces of your DNA. Okay, and they usually code for a protein, or they code for instructions for that protein. So there's some regulatory DNA in there too. And this is what makes up your chromosomes. This is what makes up your chromosomes. Okay, prokaryotic organisms, they only have a single circular chromosome with very few essential genes. They're a very simple organism. They don't need a lot of genes. They aren't going to have to make a lot of proteins. Now, some have plasmids, which are small circular pieces of DNA not on the chromosome that contain non-essential genes. And the key thing about plasmids is they can be passed between cells, which we'll talk about later briefly. This is one way that we can move genes from one cell to another, is by using plasmids. We can load a gene on a plasmid, we can insert it into a cell, and then that cell can transfer it somewhere else. In eukaryotic organisms, the chromosomes are paired, and there are many. In humans, we typically have 46 chromosomes, 23 pair. 23 that you got from your biological mother, 23 you got from your biological father. Okay, there are some humans that have maybe a few more or a little bit less, but the vast majority of humans have 46, 23 pairs. Our chromosomes are huge compared to the prokaryotic chromosomes because we have many, many more genes. This is a karyotype of a human genome. The genome is a copy of all the genes, and the karyotype is a picture. So what they do is they interrupt a cell in metaphase and mitosis, and we'll learn about metaphase in a little bit. They extract the chromosomes, they dye them so that now they can match them. They do this all digitally with a computer, so they can move them around, cut them, paste them, so they can actually take a mixture and put them together and show the pairs. And this is showing, 22 autosomes and then the sex chromosomes. And because these are the same, even though one's bent, they're still the same, this would be a human female karyotype with 2X chromosomes. Okay. And again, by using fluorescent stains, it makes it easier to highlight and match up where they're the same. Now, of those 23 pairs, 22 were homologous. 22 were homologous. And what that means, homo means the same, and logos refers to the location of the genes. And so of the 23 pairs, the first 22 they have the same, they have genes that code for the same characteristics in the same location on the same chromosomes. So the homologous chromosomes are the same length, they're the same shape, they have the same genes, or not necessarily the exact same genes. We'll talk about alleles in chapter 7. But again, one of these you got from the pair, one of them you got from each parent. Okay. Now, the sex chromosomes could be homologous if it's XX for a chromosomal female. If it's XY for a chromosomal male, the Y is considerably shorter than the X, and that makes them heterologous, not the same. And this is, again, showing you homologous chromosomes. They've been stained using different dyes to show that the regions of the chromosome and the information they carry is the same. Okay? And this is true for all homologous chromosomes. Now, when a cell replicates in your body, and this is true for most eukaryotic organisms, if we're making an exact copy of the cell to grow, to develop, or to repair, we're going to undergo mitosis. And mitosis is just a part of the cell cycle. The cell cycle is a series of events from when the cell is a brand new cell until the cell divides into two new cells. And it includes 3 phases. The first is interphase. And interphase is a long phase where it stretches through 3 different periods. Mitosis is relatively short. And the reason for that is because that's when the cell is susceptible to damage, and the damage that can't be fixed. So that has to happen quickly. And then cytokinesis is actually breaking that one cell into 2 new daughter cells. So if we look at interphase first, most cells are going to spend 90% of their time in interphase. They're going to start out when they first are formed after mitosis. They're going to be small. They're not going to be full size. They may not have taken on the complete role that they need to take on. And so they're going to undergo G1. And this is gap 1. And during this, the cell is growing, it's maturing, it's taking on whatever role it's going to be. Now, some cells never leave G1. Some cells are not going to replicate, like nerve and muscle cells. This is why when someone damages nerve tissue or muscle tissue, it's usually permanent. Now, there is some hope for these people now because of new technologies. We'll talk later in the class about gene therapy, the idea of being able to go in and stimulate cells to fix themselves that normally wouldn't. And we're going to talk about stem cell treatment and stem cell technology, where we could incorporate some cells that could develop into new nerve and muscle cells. But that's new technology that isn't quite perfected yet. So those cells that are never going to divide, they lapse out into GO, sometimes you'll see in a textbook. And all that means is that they're not going to actively divide anymore. Some of your cells, when they've divided enough times, they're not going to replicate anymore. And they lapse into G0 until they finally die and are removed. In S, if a cell needs to replicate, there's going to be a message that says we need you to replicate. And in S, we're going to synthesize the new DNA. We're going to take all the chromosomes and we're going to make a complete copy of them. We're going to duplicate each one. Because if we're going to form 2 new daughter cells, each one has to have a complete copy of all the genetic material. And then if we did that well, then we go into G2. And this is where the cell is going to break down the cytoskeleton, because we can't have that in the way if we're going to divide. It's going to start stockpiling all the monomers and polymers and extra organelles and membrane it's going to take to physically become 2 cells out of 1. And yeah, that's that. And then we're ready for the mitosis phase. Now, when we copy the DNA, okay, so if this is just a cartoon, but this is our pair of homologous chromosomes. One came from dad, one came from mom, or this is from mom, this is from dad. It doesn't matter. But when we copy them, they're not chromosomes anymore. They're going to be called sister chromatids because they're going to be attached to each other at a point called the centromere. And they're going to stay attached to each other so they don't get lost. It's kind of like if we could make a complete copy of you and you stayed attached to your twin at the hip like Siamese twins. Okay, the same thing's happening to our homologous chromosomes. And then when they finally separate, each one of these is going to go to a different daughter cell. Okay? Now, the cell cycle is controlled. Okay, we want to control it. We don't want cells to divide out of control or when we don't need them to divide. That's what happens in a cancer, and we'll talk about that in part 2 of this chapter. All right, so there's going to be a checkpoint protein that's going to make sure that we actually need this cell to divide. And if it does, then we copy the DNA, and there's another checkpoint to make sure the DNA has been copied correctly. If not, enzymes can try to fix it. If they can't fix it, then we can go through a process called apoptosis, which is programmed cell death, and we remove this cell. Because if we can't fix the damaged DNA, there's no sense in going any further. The cell is toast. All right. If everything's okay, then we go into G2, where there's another checkpoint protein to double check to make sure the DNA is not damaged, to make sure we've got all the extra organelles and membrane and nutrients that we need to make sure the cytoskeleton is broken down. And then if that's okay, we go into mitosis. So we're controlling the cell cycle with special proteins. Okay. Now mitosis, even though we think of it as 4 characteristic phases: prophase, metaphase, anaphase, and telophase. The truth is, it's not like you're in prophase and chunk, you're in metaphase and then chunk. It's like when you were first born, you were a baby, right? And you grew and you developed, and then all of a sudden one day you're wandering around, you're a toddler. It wasn't like one day you were helpless in a bassinet and the next day you're wandering around. Gradual change, and then a gradual change into a young child, and then a teenager, and then a young adult, and if you're older than that, into an older adult. Okay? Same thing is true in mitosis. It's a continual change. But it happens quickly because at any part of this, damage can occur to the cell, and now this is when it's susceptible. The first phase, prophase, is recognized by some characteristic events. We've already copied the chromosomes into sister chromatids. That happened in interphase in S of interphase. Okay, but we need these to supercoil. We need them to condense. If you've ever flown a kite and your kite's way up high and you got the long kite string from it, and all of a sudden the kite crashes, because kites always crash. And so you start walking across the field to collect your kite. You don't just drag the string behind you as you're walking to the kite, because it's going to get caught on something. It's going to get dirty. It might get broken. So as you're walking to the kite, you're wrapping the string up around a stick or a pop bottle, or maybe you've got a fancy kite reel. Well, when that kite string is all wrapped up in a small area, it's easy to move it without damaging or breaking it. And the same thing is true of our sister chromatids. So we're going to condense them. We're going to supercoil them. If we're going to separate these and make 2 new daughter cells, we have to break up the nuclear envelope, because we have to release the sister chromatids. So that breaks up. And then we start building something called the mitotic spindle. The spindle is a series of microtubules that are going to do 2 things. They're going to help us to separate the sister chromatids and make sure one gets into each new daughter cell. And they're also some are going to expand and elongate the cell. When everything's ready, then we slip into metaphase. In metaphase, our spindle is fully formed, and our sister chromatids are aligned along the equator, one sister chromatid per spindle fiber, attached at the centromere. Okay? And when everything is lined up and attached correctly, enzymes are going to break down the proteins that are holding these two sister chromatids together at the centromere, and then these microtubules that are connected to them are going to start to shorten and physically pull those sister chromatids apart. Once they separate, we call them chromosomes again. Other spindle fibers are lining up to elongate. In anaphase, we've separated our sister chromatids, the spindle fiber that they're attached to. Where's my pointer? The spindle fiber that they're attached to shortens, pulling them to the poles, while other spindle fibers lengthen, elongating the cell. Now, cytokinesis, actual physical separation of the two daughter cells, doesn't happen very quickly. So we're going to actually start that now. We're going to start making, and you'll see this in a little bit, what's called the animal cell cleavage furrow. We're going to start making a plant cell cell plate. Okay, so that's going to begin here. In telophase, our chromosomes have made it to the pole. They start to unwind. The spindle breaks down, and new nuclear membrane starts to form, and then we continue cytokinesis preparations so that we're ready to separate the two cells. And when cytokinesis occurs, that's when physical separation finally occurs. And the mechanism for this, again, is different between animal cells and plant cells. In an animal cell, fibers, protein fibers around the middle of the cell are going to pinch together. It's like if you've ever taken a long, thin balloon and twisted it in the middle to make two pieces, same kind of thing is occurring. You're pinching it in half. And as that cleavage furrow deepens, eventually that big cell is pinched into two. In a plant cell, microtubule-guided vesicles are going to carry cell wall components to the center on the cell plate. And there, as they fuse, they put cell wall material to the inside and membrane to the outside, so that when it's completely formed, we've got a brand new cell wall between the two daughter cells, with cell wall material outside, cell membrane on the inside. Realize that these two daughter cells in a plant cell are probably going to stay attached for a long, long time, whereas the cells in an animal cell might not stay attached. And so that's why we need a different mechanism for cytokinesis. So that's the end of part one. In part two, we're going to look at binary fission in prokaryotic cells, and then we'll look at mitosis and its relationship to cancer.